a little bit, so that can be a little bit confusing. But we have an axon that's branching, and what you'll notice is that in some cases, the branches of that one motor neuron terminate in adjacent muscle fibers, but in other cases, they can, let's say in a back muscle, for example, they can be in a muscle fiber some distance away. The net effect, though, is that when that little motor neuron, little action potential goes down, it is propagated along the axon to the terminal, all of these muscle fibers then, at least at that point, should be excited more or less simultaneously. And this would be good, again, for a postural muscle, or muscle that does not require a great deal of precision. So over on the left is the general sequence of events that we're going to be working through. That's just my attempt to summarize what we're going to be talking about. A motor neuron will excite a muscle cell. As you can see, it can be even several cells simultaneously. On the postsynaptic side, inside the muscle cell, there will be release of calcium. And we've indicated on many occasions before that calcium acts as an intracellular switch. Whenever intracellular, I can be more precise, intracytosolic calcium concentrations increase, something dynamic is going to happen inside the cell. And of course, in this case, it's going to be contraction or shortening of the muscle cell.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane.
So there's nothing new there. This is identical to our discussion of synapses in the neural unit. In the yellow presynaptic neuron there, we have an action potential emerging toward the terminus. And that green thing, of course, is a voltage-gated calcium channel. The action potential is going to cause that voltage-gated calcium channel to open, increasing the concentration on the presynaptic side of the intracellular calcium. And then following from that will be calcium-dependent exocytosis.
Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft.
So make sure you understand that the neuromuscular junction is a cholinergic synapse or junction. And of course, in this illustration, they look like the little orange and the lemon diffusing across together.
Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So, of course, we are presently tasked with describing muscle contraction. But be sure as we do that you understand all of the events involved in muscle relaxation as well. All right, in order for everything that we just saw to happen, there had to be some type of neural signal. That neural signal had to culminate in a motoneuron firing an action potential. And then you saw the release of neurotransmitter binding. By the way, you said cholinergic receptor. What type, more specifically, what type of receptor is this? Nicotinic. So in order for all of that to be terminated, each one of those stages must also be brought to a halt. Another common mistake that I see on the tests is that students often confuse what happens in the presynaptic cell from what happens in the postsynaptic cell. So to head that off this time, I put them in different colors. So make sure, I broke my computer now. Make sure that you don't confuse those two. So we're going to find that calcium, for example, has a very different role in the presynaptic cell than it does in the postsynaptic cell. And by the way, I've also started to include, as we continue our discussion, some of these abbreviations, some of these formal abbreviations. You need to know those. We talk about nicotinic receptors. I have a little abbreviation for those in just a moment. So I would expect on a test you to know what this means, for example. The symbology is in all the pharmacological and clinical literature, so just it's something you need to be familiar with. So we saw this illustration before, and in the dramatic oversimplification that is applied here, we have a we have a sort of a muscle. I guess it'd actually be equivalent, sort of a amalgamation of a muscle fibril and a muscle fiber. But in any case, then you can see the sarcolemma labeled there. You can see the transverse tubule, and the little arrows there are intended to indicate that an action potential has been propagated into the transverse tubule. And then remember we talked about the triad region being very important for signaling. What's going to happen is that AP, that action potential, is in a manner that we're going to sketch out, that's about as good as we can do, is going to communicate with the cistern region of an adjacent SR, causing those yellow things to lead. We talked about this before. The SR is a cistern for what? What is it holding? What does it contain? Calcium, yeah. So basically what we're saying is the action potential moving down the transverse tubule is going to induce release of calcium from the SR, which will then bind into this actin filament system over here, causing initiation of the sarcomere to shorten. Now, calcium is going to be very important in the postsynaptic side. When it is released, that will be, as we've indicated, that will initiate the events that culminate in muscle contraction. But I guess the point I want to make before we talk about that in any more detail, I want you to make sure that you understand there are pumps. Remember, we talked about primary active transporters before. There are pumps in the membrane of the sarcoplasmic reticulum that are there to ensure that under normal circumstances, the calcium ion concentration in the muscle cell is extremely low. So these pumps are always on. They're like bilge pumps in a boat, always making sure that the calcium is at highest concentration inside the SR and not outside the SR. By the way, calcium is also pumped outside of the cell as well. There are specialized proteins. There are several of these. One you should be familiar with, calsequestrin, to sequester something. What does it mean to sequester something? Gather and hide away. Yeah, gather and hide away is sort of the idea. So the term sort of tells you what it's doing. It's sort of attaching to it, binding it up, preventing it from doing anything else. It just sort of locks it up.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, This causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
So let's be sure we understand the precise sequence of events. I think the next slide adds a little more meat to this. No, that's not the one I want. Here. So there's an action potential that's going to be propagated down the transverse tubule. There's our transverse tubule. And I think Bob pointed out earlier, there must be some way to transduce that action potential into a form that results in the release of calcium from the SR. Well, lining that transverse tubule are what are called DHP receptors, dihydropyridine receptors. And as it would turn out, we didn't know this until recently, these are nothing more than specialized calcium channels. And they're going to turn up again later, so I think you probably ought to know them now. You're going to have to learn these later anyway, but these are L-type slow calcium channels. L-type slow calcium channels. And they seem to open, they seem to be sensitive to a change in membrane potential, and that's what the action potential was doing, changing the local membrane potential, causing calcium-dependent release of calcium. Calcium-dependent release of calcium. So this calcium channel is going to allow a small amount of calcium to enter the cytoplasm. And we're going to treat this as ECF now. This thing, this voltage-gated channel, is a calcium channel. Calcium is higher concentration outside the cell, comes into the cell. But just enough to trip another receptor, this time located on the membrane of the sarcoplasmic reticulum. So remember, calcium acts as a switch. The intracellular calcium now concentration has changed sufficiently to cause this receptor, which is usually called a ryanodine receptor, RYR, in the SR membrane to open. And of course, we know the calcium is located in there. Now, we're going to come back to that in a moment. But the function of the calcium in this story is to bind to troponin. Calcium binds, forming a complex, altering the conformation of the troponin, displacing the tropomyosin, which you can see runs along with the F-actin filaments. And when the tropomyosin is moved, when the conformation changes, the myosin cross-bridge head can now attach to the actin filament. One very important step in our process of sarcomeric shortening.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
So as you watch the sequence of events repeating here, according to the narrator, ask yourself or be able to tell me what is the function of calcium? What is the function of calcium in this context? Or the calcium binds the troponin, but to what end? All right, so that when the calcium binds the troponin changes conformation, changes shape. When the troponin changes shape, because it is bound to the tropomyosin. The tropomyosin changes shape or conformation. All right? Why is that important? When the tropomyosin changes shape or conformation, what can now happen? The myosin cross-bridge head can now attach to the actin. Remember that process of attachment is necessary for the walk-along process to occur. All right, so as you looked at the illustration or looked at the animation, you established or we found that calcium is dependent for attachment and contraction. What's then the role of ATP? I would say there are two important roles. What are they? For what? Huh? Exactly right. So ATP is necessary for two important functions: conformational change of the myosin cross-bridge head, that when it's attached and it changes conformation, that's called a power stroke. The power stroke, of course, is necessary to draw the myosin filament along the actin. That's the power stroke. So ATP is necessary for that indirectly. I remember we said earlier that the myosin cross-bridge head is a hydrolase. It's an ATP hydrolase. It has the ability to extract the energy in the phosphate of ATP and use that for some purpose. Well, one purpose is to change its conformation, essential for the power stroke. And there's one more thing. What happened when the ATP attached? It detached. All right. So strictly speaking, ATP is also necessary for detachment of the myosin cross-bridge head from the actin. And this will continue to a large extent in a calcium-dependent fashion. As long as the calcium ion concentration in the cytosol is sufficiently high, this process will continue on and on and on. Again, be thinking about what is necessary, what would be necessary to terminate all of this, because that has to happen every time that you make a rapid movement. You're playing bongos. Anybody play bongos anymore? You're playing a guitar, whatever, very rapid muscle movements. That means that all of this has to be cycled on and off with the rapidity with which you're moving and stopping the movement of your fingers. By the way, I didn't mention this, but an additional cofactor necessary for muscle contraction is magnesium. We won't get into the role, but I would say that over half of the metabolic reactions in the cell, all the cells of your body, involve metallic cofactors, everything from magnesium, manganese, iron. All of these are sometimes used. All right, so let's go back to this illustration. This is a ridiculously silly oversimplification, but it does help us to introduce some of the figures we haven't talked about too much. So we talked about the AP coming down the T-tubule. We talked about the importance of the DHP receptor. The DHP receptor is what? How would you describe the DHP receptor? What is it? All right, so basically it's a calcium channel. It's a slow type calcium channel that we're going to encounter again when we talk about the heart. But more importantly, all right, so it's voltage gated. So this is a voltage gated calcium channel. And do we understand this concept of calcium-dependent calcium release? Remember, calcium is the switch. And I've got a little switch here. This is my little switch. You know what a relay is? In your car, you probably had to replace a relay in your car. What does a relay do? It takes a small current, and then it, I'll give an example. Have you ever played around with the car battery? It's just something to do this afternoon. And you can hook the car battery up to the fillings in your teeth or whatever, do all sorts of things. You can weld with a car battery. But let's say you just wanted to turn your radio directly with 12 volts. Look, that's not the best example because you won't drive current into it. But if you just take a small switch, let's say you go to the hardware store and buy a small switch, and you want to put it across the car battery because you found that there's a giant spark every time you take your screwdriver from the positive to the negative, and you don't want that to happen anymore, so you go down to the hardware store and you get a little switch and you put it across the terminal. What's going to happen the first time you flip that switch? It's going to melt. All right. So what could you do to prevent that? You could use something called a relay. You could take a very large set of conductors that can handle that high current, and and then use that to turn on, let's say, your radio or your headlights in your car. And that's what you have relays in your car for. Now, a lot of them are electronic, but that's what a relay does. Same idea here. A very small current is used, a very small signal is used to generate or turn on a much, much larger calcium current. And this is the one that's going to trip the sequence of events we just talked about in the animations. The attachment of the calcium to the troponin, the change in the conformation of the troponin. Troponin moves, the tropomyosin moves out of the way, allowing the myosin cross-bridge head to attach. And then we have the ATP-dependent change in conformation and detachment following each power stroke of the myosin cross-bridge head. I like the way that they have cast this ryanodine receptor as a trap door. It's a spring door, a little spring-laden trap door. And of course, when it opens in response to the slight increase in intracellular calcium, the calcium just pours out of the SR, allowing this process to continue. And then, once again, if you want to bring this to a grinding halt, we need to stop this immediately. What has to be done? I want to relax the muscle now. I'm tired. All right, and so we talked about what Bob said, get the calcium back into the SR. What does that? Make sure you know it for the test. The Cerca pumps, SERCA, the Cerca pumps that pump it back into. These are very, very, it's like the airbag in your car. In fact, if you're playing around with your car battery tonight, there's probably a way you can get this airbag to go off too and demonstrate this procedure as well. But in any case, the very, very rapid transport of calcium. And we also indicated that calcium is being pumped out of the cell as well. It's pumped back into the SR, and because you've got to get that intracytocellular concentration down into the nanomolar level again. Yes, sir.
Are those pumps on your slide that you just mentioned?
Are they there?
I cannot see them.
You're asking, can you see them?
I cannot see them.
You cannot see them.
Okay. So, can you spell them again for me, please?
Oh, they're back here. SERCA. Sarcoplasmic, what do they stand for? Sarcoplasmic something, calcium, I can't remember the abbreviation anymore, it's so late in the day, but here they are. That's what we talked about before. Yeah.
Thank you.
That's the model we're going to use. For our purposes, this sequence of events will only work if you think about the T-tubule being filled with ECF. So essentially, the T-tubule is bringing the exterior world into the interior of the cell. But I agree, I do not have a good picture of the Cerca pump. And we talked about relaxation. These are just a few of the events that are necessary for muscle relaxation. You can probably add a few more points to this, so be sure you can do that. Any questions then about the general sequence of events involved in muscle contraction? Be sure you know what we mean by RYRs. And there was another abbreviation I wanted you to know as well. Notice the scientific representation, the scientific symbology of nicotinic receptors. There are a whole bunch of different types of nicotinic receptors. We're not worried about that. We just want to, when you see that abbreviation, you should know what we're talking about.
So,
yeah,
the calciquestion binds to the calcium in the sarcoplasmic.
In the SR.
All right. So, what makes it unbind so it can be used after the vanadine receptor opens?
Yeah, the prevailing concentration. So if you're asking what causes let me think of it this way, a sponge. Let's think of the calciquestrin as a sponge. What causes the sponge to soak up the water? That's not a metaphysical question. I'm not asking grasshopper or anything. What causes the sponge to soak? What enables the sponge to bring up the water?
Expansion.
Will a dry sponge, this is where the grasshopper comes in, will a dry sponge bring up the water?
Yes.
If there's no water, just wait, it's been all day. If there's no water, will the sponge attract the water. Can the sponge, can we create water if there's no water there? So my point is, what I'm trying, it's just stoichiometry. Remember in your chemistry, the stoichiometry, the prevailing calcium concentration, the calsequestrin is only going to bind the calcium if there's a sufficiently high concentration. It's being pumped into the SR. That's going to facilitate stoichiometrically binding of the calcium. If we now open those ryanodine receptors and calcium concentration dramatically lowers, there's less propensity, there's no more water to go into the sponge.
Okay, so when it's already bound, it just automatically releases just
And
because
well, that's true. That's true for a lot of.
Okay.
That's true for a lot of protein binding. It's all going to be driven by concentration. And by the way, there are many other types of calcium binding proteins, both in your blood and in other cells. There must be at least a half a dozen different calcium binding proteins. We'll talk about a few more as we continue in the course. We don't have a lot of time to talk about what I've called here whole muscle contraction, but I do want to mention a few general concepts related to the energetics of whole muscle contraction. So first of all, understand how we're changing our focus. We're moving away from individual cells and fibrils and filaments and so on. We're talking about entire muscles. And we've indicated that ATP is necessary to drive all this. We've talked about calcium, but of course the energetics are very much dependent upon adequate supplies of ATP. So where does that ATP come from? You may have seen, maybe some of you are using this. Any of you got a Jones for some creatine phosphate? Huh? So what do you use it for? All right, and what are the putative benefits of this? What is the idea behind taking it? What? Okay, and why would that be? Because what I'm asking for is, you're exactly right. Both of you are exactly right. Let me put it this way, and then you tell me why this is happening. If I were to take a little meter, let's say I have an ATP meter, and I'm going to take one end of my ATP meter and put it inside your muscle cell, and I'm going to measure the concentration of ATP as you're contracting your muscle, contracting, relaxing. What would you expect to happen to the ATP concentration as you're contracting and relaxing as I measure it with my ATP meter? It's going to fluctuate, right? It doesn't. Why? Why does the ATP remain in a fairly consistent concentration independent of activity? All right, because there's an enzyme that has the ability to transfer very rapidly, only for a very short period of time. You can see on the order of maybe 10, 20 seconds. Phosphate, which is the dollar bill, whatever analogy you want to use, it's the dollar bill, it's the fire, it's the battery of the cell. But it's able to transfer that very rapidly, kinetically, extremely rapidly to the ATP, maintaining the concentration. So that this process, this ability to maintain ATP concentrations inside the active muscle cell is dependent upon adequate concentrations of both creatine phosphate, or phosphocreatine, however you want to pronounce it, and the enzyme up there, the creatine kinases. Remember, kinases in general are enzymes that facilitate transfer of phosphate from one molecule to another. We'll talk about these more later. So this is sort of your short-term energy helper. Any downsides in the literature? Those of you who haven't ever seen it? Yeah, there was some early reports about increased risk of tendon tears and muscle strains. I don't know if that's all been supported, but it seems to have at least so far, no major adverse consequences. All right, so that's only good for about 15 seconds. Then, of course, skeletal muscle has fairly high concentrations of what is called myoglobin in it. There's a muscle myoglobin that's a little bit different than the hemoglobin in the blood. That will help. You can also derive glycolysis. You can derive energy from glycogen that's stored in the muscle. This will go perhaps for another minute or so. depending upon, again, the intensity of the exercise, how rapidly, how heavily you're breathing, and so on. So, in general, and I think you've established this, we've even had a quiz question related to this, aerobic respiration, if you're engaged in, if you're depending upon your genetics and a variety of other factors, if you can maintain a fairly intense level of activity with aerobic respiration, you've got the best of both worlds. There's this, read about this Mexican bicyclist. Anybody read about him? He's maybe favored to win the Tour de France this year. Is it Mexican?
No, Colombian.
Colombian, thank you. And they measured his VO max, and apparently it's one of the highest. He's a small fellow, he's not large at all. They measured his VO max, and it was greater than most anyone else they had measured. I think they said it was equivalent to Lance Armstrong or something. We all know about Lance Armstrong. But in any case, what that means is this fellow is able to maintain aerobic respiration at a very high level, high intensity. And of course, if that's possible, then you don't have to go to the short-term glycolytic stores. What tends to happen in skeletal muscle fatigue is the nervous system sends signals to the skeletal muscle, but it no longer obeys those signals for largely chemical reasons. There's an insufficiency of calcium, there's an insufficiency of ATP, there's an insufficiency of critical enzymes, just perhaps an insufficiency of oxygen availability. But whatever the reason, the muscle no longer responds to the neural signals. We've talked about the relationship between sarcomere length and the force of muscle contraction. You sometimes see in the current literature, I don't really care for the use of the term this way, but the term preload is sometimes intended to represent warming of the muscle. By the way, don't confuse stretching. We've had a question in your little quiz today about stretching with warming up. Nobody disputes the importance of warming up prior to exercise or athletic performance. But there is, especially if you're into weightlifting or resistance exercise, something called preloading. And the idea there is that at the molecular level, not at a kinesiological level, but at a molecular level, you're getting those sarcomeres at the optimum length through a series of preliminary exercises. The term afterload in the context of skeletal muscle activity refers to the force or tension that's needed to actually lift a load, displace a load. And the general premise is the lower, the less the resistance, the smaller the weight, the more rapidly you can move it. That shouldn't violate any principle of common sense. We often differentiate isometric and isotonic contraction. Isometric contraction implies that the muscle is under tension, but it does not displace the load. So the sarcomeres, the fibrils are utilizing energy. They are attempting to contract, but they're not displacing the load. Isotonic contraction, on the other hand, implies that the sarcomere, as it shortens, is also displacing the load. You actually have something called eccentric contraction. What is eccentric? This is called contraction. What is eccentric contraction? Anybody familiar with that? Yeah. Explain. What would you do?
It's like doing a push-up that's you're doing bench press and letting it down the eccentric phase.
Exactly right. You're basically in eccentric contraction. The load has exceeded your ability to displace it. So that if you're doing a pull-up, you may start here and you're trying to pull yourself up, but you go down. Or as you described, the push-up, you can no longer push yourself up. Again, in Evan's world, this is intended. This is intended to facilitate the temporary destruction of the muscle tissue, so it can become more hypertrophic later. All right, a few more terms that we should be familiar with. By the way, this little introductory point here I think is fairly self-evident. If you are sending signals to the muscle to contract, in general, it is the case that the higher the frequency of action potentials, the greater the force of contraction. So that's one way that the force of contraction can be increased. Force of contraction would vary with the frequency of action potentials. What else would increase force of contraction? I'm sorry? Explain. We're going to talk about this. I just want to.
Each motor unit innervates specific muscle fibers, so if you recruit more units, you're recruiting more fibers.
Exactly right. So, and the general principle is when we're picking something up, we use the smallest motor units first, and then as we need to increase the force of contraction, we recruit, that is, we add larger motor units. So there are a number of ways that force of contraction can be enhanced by frequency of action potentials, by recruiting additional motor units, and so on. Remember our definition of motor unit. The term twitch just is sort of an idealized term. It has no objective reality. It's sort of a physiological fiction. But here we're talking about just a single contraction. We'll just call it a single contraction. Generally, you could think of it resulting from a single action potential, although that might be a little silly. The term recruitment just means a progressive addition of motor units to increase the force of contraction. We'll talk about how this can be described in terms of summation, as we talked about it in the nervous system. This is an interesting phenomenon you should be familiar with called Treppe. It's a German concept. Treppe means staircase. And it's a little bit problematic to get a handle on, but again, let's say we're doing those curls again, and you pick up the weight. Let's maybe pick up a little weight here, and I don't know. Let's say we have some type of meter that measures your comfort or your relative ability to lift that weight. Or is it going to be easiest for you with the first contraction, or maybe with the fourth or fifth contraction? Which is going to be easier, the first time or the fourth time? Not a heavy weight, just let's say 20 pounds, 15 pounds, 10 pounds. Which is going to be easier, first time or third or fourth time? Yeah. Treppe says that with successive low-frequency contractions and relaxations, and that's the critical point, and relaxations, the force of contraction increases until what happens? The muscle becomes fatigued, rather like all of us today. Right? So Treppe says with each low-frequency contraction and relaxation is the critical point here, because we need to differentiate this from fusion. Force of contraction will increase until muscle fatigue sets in. So if we have some type of dynamometer, a dynamometer is just going to measure tension over time. And we're looking at this top left illustration here. We can see each one of these little triangles at the bottom represents a stimulus. This could be your brain telling your muscle to contract, or it could be something like a transcutaneous electrical nerve stimulation device. Anybody have one of these at home?
Sure.
You use them? The PT use them a lot. Yeah, what do you do with it? Well, they can be used for that purpose, among other things. Yeah, you put it on there. None of you use these for weight loss? Yeah, exactly. Yeah, as I say, the PTs use them quite a bit for even thermal. They do all kinds of things. But often you'll have patients who don't have a lot of muscle control, and you can artificially sort of induce contraction with these devices. But that's what they do. They induce muscle contraction in a intensity and frequency specific fashion. So I have two dials. Let's imagine I have two dials on my TENS device. And one is magnitude and the other is frequency. All right. So if we go back over here to this illustration, this is what, 100 milliseconds, 200, 300, 400. So how many milliseconds in a second? Yeah, 1,000. So the 200 would be about a fifth of a second. You can see that third one there is about a quarter of a second, and so on. So the thing on the left is a twitch, and you can see it takes about, look at that, over a tenth of a second to initiate and then dissipate. Now, we've seen something like this before. How would you explain this? Now, this is not neural, this is muscular. This is a muscle contraction. What's happening there? Yeah, we're going to give it a slightly different name, but it's exactly the same thing as temporal summation. A muscle is induced to contract, and before it has time to relax completely, it gets another stimulus, perhaps via the TENS device. And so the net result is what? We get a higher force of contraction, a greater force of contraction. Now, what is the difference between this and this? I'm getting increased force of contraction here, and you would agree that I'm getting increased force of contraction here as well. Exactly right. So make sure you understand that in TREPA, as opposed to frequency summation, we have contraction and complete relaxation. Contraction, relaxation, and then there will be a progressive, if mild, increase in force of contraction once again until fatigue sets in. Over on the right here, each one of these represents a stimulus. Is it clear to you how we're getting that frequency summation? Why isn't this TREPA?
No
It
relaxation.
doesn't go all the way down, doesn't relax. And then at the top here, what you'll see is eventually that muscle reaches maximum force of contraction. You see this in wrist wrestling championships a lot. You probably don't watch as many wrist wrestling matches as I do. But frequently, the wrist wrestlers will do this toward the end. Or you'll see this, all kinds of things you can do. That's called unfused tetanus. Now what you'll notice is that the muscle has peaked out. It's reached its maximum force of contraction, but it oscillates, it vacillates back and forth a little bit. Unfused tetanus, compare that to what's happened down here. Boom, muscle locks into tetanus and remains there. There's no vacillating. And if you look at these two illustrations, what's the primary difference?
Amount of
The frequency of stimulation.
stimulation.
Frequency of stimulation. This occurred so rapidly because each one of these little triangles represents a higher, well, relative to this, a higher frequency of stimulus. So do we all have a handle? We can all differentiate, at least on a biograph like this, the difference between a frequency summation and a TREPA.
Mm-hmm.
So what's going on here? That would be frequency summation. What do we have up here? Well, how would you classify this tetanus? Fused tetanus, right? Everybody see the distinction? All right. So what we'll do, we'll stop it there. I don't know why my little animation didn't work there. Or it says it cannot play the music. It's a dancing man. But in any case, we'll stop there and then next time begin looking at smooth muscle.